So now we can go ahead and now look at the whole process of mitosis. I think that was the goal and the gist of the whole matter. Going ahead to look at how the different the cells, how the chromosomes behave during mitosis. As we said earlier, mitosis usually involves the cells dividing, equally dividing into two identical cells. Okay? And this one involves the chromosomes, and those cells are identical simply because they contain the same number of chromosomes. If the parent cell was 46, the daughter cell should also be 46. And we achieve that simply because we are able to undergo what they call replication of the genetic material during the S phase of interphase. So when you look at the prophase, when you look at the whole process that is involved in this mitosis, I can give you an acronym that can help you remember the processes, the stages in their rightful order. That is to say, PMAT. Interphase is not part of mitosis. Interphase is preparation stage. Mitosis starts from prophase, metaphase, anaphase, and telophase. So during prophase, we are going to go ahead and look at the different events that take place during each of those stages, and then we shall go ahead and and that will be done. So we are going to start with the interphase. Okay, when you look at interphase closely, you realize that now originally the chromatids usually appear to be very small and thread-like. That is during interphase. That's interphase. You can clearly see that. So whenever they ask you about mitosis, mitosis does not have interphase. Interphase is a preparation for mitosis. Okay, so so now when you look at during interphase, most of the types, we can just recap ourselves what we usually we did in mitosis. Cells is actively synthesizing proteins. The chromosomes are not even visible. They are thread-like. When you put them under the microscope, they are very thin thread-like structures. The DNA also replicates. You can see that the nucleolus is intact and the nuclear envelope is also intact during the interphase stage. Now, after interphase, what we go to next is what we usually call prophase. Now, during prophase, the chromosomes which were originally thread-like, they condense into clear chromosomes, as you can see clearly from here, that the cells, the chromosomes have condensed. They have condensed into chromosomes, which are made up of two chromatids, sister chromatids. One of the two structures that make up the chromosome, a sister chromatid. A chromosome is made up of two sister chromatids, which are identical to each other. Why? Because they were formed during the synthesis phase of interphase, the S phase of interphase. Now, during prophase, that is when we start seeing the nuclear envelope starting to break down or to disintegrate. Some books will tell you that the nuclear envelope starts to disassemble, disintegrate, break down. And then we start seeing the nucleolus starting to disappear. But you can also see some structures known as the spindle fibers. Some books will call it microtubule assemblage centers. You can see that the spindle fibers have started to form, coming from the microtubule arrangement centers. The microtubule arrangement centers, they are the centrioles. You can see the centrioles. That is where the spindle fibers originate from. Though plant cells do not have centrioles, so centrioles are not so, their function is not so clear. But we should note with me that the chromatids are still connected to each other to form a chromosome at the point known as the centromere. Now, events to remember: chromosomes condense to become visible. 2. Nuclear envelope starts to disintegrate. 3. Nucleolus starts to disappear. Spindles start to be formed. Now, that is prophase. They usually call it prophase simply because it is before, it comes before the chromosomes align themselves at the spindle equator. That is pro. That is why they call it pro. Now, we go ahead and look at our next stage, and that is metaphase. Now, during metaphase, there are a number of things that we usually note. In metaphase, you can clearly see from the screen that now all the chromosomes which were scattered all over. Remember, in prophase, we have lost the nuclear envelope, we have lost the nucleolus, but we ended when we had our spindle fibers forming. So the spindle fibers are now going to go ahead and attach themselves to the chromosomes, but they attach themselves to the chromosomes at the centromeres. The spindle fibers are going to attach themselves to the chromosomes at the centromeres. And two, the chromosomes are all aligned at the metaphase plate in the middle of the cell. That is to say, the metaphase plate. That's why they call it metaphase, because the chromosomes are aligned at the spindle equator, all of them. And now the spindle fibers have made contact with the centromeres. Now that marks the end of metaphase. Now anaphase. I like remembering anaphase by remembering that ana away. So the centrioles start to you can see that now the spindle fibers are starting to contract. The number of proteins that are usually present in the spindle fibers, but the spindle fibers start to contract. So the spindle fibers start to contract. As they contract, because they are contractile in nature, they start pulling apart the sister chromatids. Remember, the sister chromatids were held together by the centromeres. So what happens is the sister chromatids start to be pulled apart from each other. So the centromere has been torn apart because of the centrioles, which are now because of the spindle fibers attached to the chromatids. They start pulling them towards the opposite poles. So when they start pulling them towards the opposite poles, then that marks anaphase. Away, anaphase, away, metaphase, middle, prophase before the middle. Remember those things very well. PMAT is our acronym to remember. So now, anaphase, they have all been pulled apart towards the opposite poles. We shall also interpret a graph, God willing. I think we have a graph to interpret there. We can look at a data interpretation and how they are formed. So now anaphase marks the end of anaphase ends when all now they have finally reached the poles of the chromosome. When they have now, the moment these sister chromatids separate at the centromere, they no longer called sister chromatids. They take on the name known as chromosomes. You will no longer refer to them as chromatids, but you will always now refer to them as chromosomes. Remember, during interphase, they were chromosomes, and now they divided. Each chromosome formed another one, but they were still connected together. So since they are still connected together, they are still being called sister chromatids. But the moment they are pulled apart towards the opposite poles, they recover their name, and now they are being called the chromosomes. Now, it marks the end of anaphase when they have finally reached the opposite poles. And now, after the chromosomes have reached the opposite poles of the cell, that marks the end of anaphase. And that marks the beginning of our next stage known as telophase. So during telophase, we usually see now the chromosomes have reached the poles and they have become distinct. They have reached the poles and they become indistinct. They start to uncoil. Remember, for the chromosome to be distinct, you have to condense, like what we did during prophase. They were condensing so that you can see them under the microscope. So the chromosomes now are going to uncoil and become indistinct. You cannot easily observe them. Now, the nuclear envelope that disappeared during prophase is going to reappear. The nucleolus is going to be reformed. You can clearly see that the nucleolus has been reformed. You can clearly see that the nuclear envelope has been reformed. The spindle fibers, all the spindles disintegrate. And after that has all happened, the daughter cells have, the daughter chromosomes have reached the sorry, the daughter chromosomes have reached the opposite poles. The nuclear envelope reappears. The nucleus reforms. The spindle fibers disintegrate. That marks the end of telophase. Now, after mitosis is done, then that is when now cytokinesis will come in to split these cells into daughter cells. So cytokinesis is like the stage that comes after mitosis. So that is briefly what we usually talk about when when we are talking about the different stages. So here is an outline of the things that usually happen during each of the stages. We have talked about the major stages that happen during each of these stages. We have said that during prophase, that is, the cells, the chromosomes become clear, condensed, shorten, and thicken. That is when you will be able to see them. The microtubules, they start developing, and the spindle apparatus starts to show. We said also that the nucleus disappears. The nuclear envelope breaks down. That leaves the chromosomes free in the cytoplasm of the cell. Now, we went ahead and said metaphase, that is in the middle. We said the chromosomes are arranged in the spindle equator. That is, say, they remain attached to the microtubules at their centromeres. Anaphase, we said during anaphase, the centromere divides into two, and then the microtubules that are attached to each, they contract. As they contract, they pull the sister chromatids that makes up the chromosome to separate them and move to the opposite poles of the cells. We went ahead and said the chromatids are now known as daughter chromosomes. They now move to their respective poles. Now we went ahead and said during telophase, now the daughter chromosomes reach their respective poles and they become longer and thinner, disappearing altogether. Now we went ahead and now they are now being called chromatins. We went ahead and said that the nuclear envelope reappears, the nucleus reappears. So that is basically what happens during mitosis. Maybe if we are to go ahead and talk about the importance of mitosis, mitosis is very, very important in a number of things. Mitosis is very important when it comes to ensuring that the cells grow. We are able to grow simply because of mitosis. We are able to replace our cells simply because of mitosis. We are able to repair the tissues that are damaged. You might be there wondering how, for instance, someone who is cut and has a wound, how they are able to repair themselves. That is mitosis. Exact identical cells will divide and then form other cells to come and close up that area. In asexual reproduction, you've heard about propagation, vegetative propagation, where a stem cutting is used to form to be as a planting material, like in the cassava stem cuttings. That is all mitosis at work. Now, having looked at, so that is basically the importance of mitosis. We have looked at all that. Yeah, so basically that is it. You'll see applications of mitosis. You will see applications of stem cells. Yeah, I think we can do one question and then we call it a day. Is it fine with you?
Yeah.
Okay. So we can try out this question. Just a moment. Yeah, we can try out this question. We can try out this question.